Good morning or good afternoon, everyone. I'm Julia Caro, Senior Editor at GenomeWeb, and I'll be your host today. The title of today's webinar is NGS Roundtable, Ask the Experts About the Future of Clinical NGS Testing. The sponsor of this webinar is PerionDX. Our roundtable participants today are Dr. Ali Grossman, Assistant Professor of Pathology at the University of Utah and Medical Director of Surgical Pathology and Molecular Oncology at ARUP Laboratories. Dr. Carl Volkerding, Professor of Pathology at the University of Utah and Medical Director of Genomics and Bioinformatics at ARUP Laboratories. And Dr. Rakesh Nagarajan, Chief Biomedical Informatics Officer at PerionDX, who will be moderating the panel discussion. Dr. John Pfeiffer from the Washington University School of Medicine unfortunately cannot join us today due to inclement weather that interfered with his travel. You may type in a question at any time during the webinar. You can do this through the control panel, which normally appears on the right side of your screen. Click on the Q&A box on the upper right side of the control panel, and when you click on Send to, please select All Panelists. Today's webinar will have an interactive format driven by questions that you, our audience, provided through a pre-webinar email survey, as well as questions you enter through the Q&A box. We have structured the event as follows. Rakesh will introduce our first topic, Emerging Scientific and Clinical Trends in NGS Testing. And Ali will then give a short presentation about liquid biopsy testing for cancer, as well as next-gen sequencing to predict immunotherapy response. This will be followed by a panel discussion based on your previously submitted questions. After that, Carl will introduce our second topic, NGS Clinical Lab Operations and Quality Management. And our panelists will discuss that topic based again on questions you submitted through our survey. Following this, I will moderate a short Q&A session to address additional questions submitted during the webinar. I will now hand it over to Rakesh. Please go ahead.
Thanks, Julia and GenomeWeb for hosting this webinar. It's a true pleasure moderating this roundtable session with professional colleagues of mine who are real leaders in the cancer and constitutional clinical genomic testing space. We've organized this webinar into two segments, one really a scientific and clinical segment focusing on emerging trends, and a second regulatory and quality management issue segment, both obviously tying to next-generation sequencing-based testing. Without further ado, I want to introduce Ali Grossman from the University of Utah and ARUP Laboratories, who will kick off the first session where we'll be discussing NGS-based testing to detect cell-free DNA and to make immunotherapeutic decisions in the oncology setting. Ali?
Today, I would like to emphasize right off the bat that what I'm going to talk about is really emerging testing in the solid tumor oncology field, and I would like to emphasize this is emerging, and very few of the tests and platforms and strategies we're talking about today are actually in the clinic. A couple of them are. The other thing I want to just acknowledge is my colleague, John Pfeiffer, who couldn't be with us today. He helped compose the content here and had a lot of input, and I'm sorry he can't be with us. But I'll do my best to represent his input. So first, liquid biopsies. So we're talking about testing for mutations in solid tumors with cell-free circulating tumor DNA. This is a really exciting topic with quite a bit of potential clinical utility. I'll try to touch base on really very basic general strategies, what the limitations are right now and what they might be in the future, and who are the patients that might benefit the most. Next slide. So first, what is the potential clinical utility of liquid biopsy? And I want to emphasize the word potential. There are several. This could be used, so taking a blood draw from a patient can be used to establish therapeutic targets, to detect residual disease after therapy, meaning monitoring treatment response, to detect relapse or progression, detect development of resistance mutations, and potentially even limit more invasive, higher risk, more costly procedures, potentially limit costly high-resolution imaging. Go ahead, next slide. What might be some strategies for interrogating these somatic alterations that we might find in the blood? There are two general strategies. One is you can go after very specific known mutations with a targeted mutation detection. And let me emphasize there are a lot of different methods to do this, and each have their strengths and weaknesses. I've listed just one here that is popular right now and has made it into the clinic, and a technology that we use here at ARUP. It's called digital droplet or emulsion PCR. The other strategy would be to look at a whole genome, a whole exome, or a broad or even a small hotspot panel, but multiple targets with an NGS panel. And I've listed just a smattering of references for our audience. There are a lot of papers on this topic, and the papers listed here are by no means comprehensive. Next slide. It's really important. I think this is probably one of the most important slides here. It's really important to talk about limitations of liquid biopsies. And probably the most important concept is when using NGS panels to interrogate multiple mutations from liquid biopsy specimens. There is some really special bioinformatic pipelines that are going to be required for this, and these are emerging, and I would refer our audience to a recent paper in Nature Biotechnology that I have listed here. The group from Stanford has an excellent publication, but this concept of using barcodes to eliminate false positive from sequencing artifact is really important. These are emerging bioinformatic and nucleic acid chemistry approaches, and we're going to see more and more papers on this. There are some publicly available pipelines and software tools that can be used for this, but again, this is emerging. And another concept that's really important in terms of limitations is the sensitivity. The sensitivity at this point, in general, appears to be less than testing the solid tumor directly. I think that's going to change over time, and I think sensitivity is going to improve, but right now it appears to be less sensitive overall. But that's going to depend on the tumor burden of the patient, and we see this in multiple publications and multiple studies where, depending on the tumor burden in the patient, your ability to detect very low frequency mutations in a cell-free DNA fraction can vary widely. Next slide. So where might this have the most clinical utility? I think that's going to be in lung cancer, and the reason is because lung cancer has multiple actionable targets, and yet we are required to test for these targets on very limited tissue from lung biopsies. So you have multiple targets that need to be interrogated, and they compete with each other. In addition, pathologists need the tissue for a diagnostic workup. So some of the tissue gets used on immunohistochemistry, for example. And when that's established, then mutation detection and biomarkers for immunotherapy begin. And so right now, EGFR and ALK are part of the standard routine testing for lung cancer. However, there are many other mutational actionable targets to go after that are emerging with actually drug data linked to them. And then recently, there's been a big demand for PD-L1 biomarker testing, and these are immunohistochemical stains. And there are 2 FDA-approved biomarker assays for PD-L1 from 2 different companies that go with 2 different drugs. Recently, in this fall, the FDA approved the drug pembrolizumab, Keytruda, in the first-line setting for lung cancer. So this is for lung cancer that is EGFR and ALK wild type. And at this time, this is the only immunotherapy that's FDA approved for newly diagnosed lung cancer patients. And there's been a huge surge in test ordering for the companion diagnostic that goes with that, and that is the PD-L1 22C3 immunohistochemical stain. And again, this competes for tissue with all these other markers that need to be tested. And it presents a real challenge for the proceduralists, the radiologists and pulmonologists who are sampling the biopsies. There's a demand for them to get more tissue, which is hard because they're a risk for the patients. It puts a lot of pressure on the pathologist who has to very carefully manage this tissue to accomplish everything the oncologist needs to take care of the patient. And that brings up the topic of immunotherapy. Next slide. So what's really exciting right now in oncology, and I'm sure you've all have heard about this, is immune checkpoint blockade for cancer patients. And I think probably the 3 tumor types where this has been most of the data is in melanoma, lung cancer, and renal cell carcinoma. So there are 2 pathways that have FDA-approved drugs on the market. I'm sure you've all heard of this. So the CTLA-4 pathway and the PD-1, PD-L1 pathway. And I'm not an immunologist, but I can just briefly give you an overview. So the CTLA-4 inhibitors, like ipilimumab, interfere with T-cell activation. And so antigen-presenting cells, so APCs, present tumor antigens, novel tumor antigens, to a T-cell, and the T-cell becomes activated to attack those tumor cells that are expressing those antigens. And this is where mutations come up. Somatic mutations create what's called neoantigens, so tumor antigens. So once that T-cell is activated, it's going to be searching out those tumor cells to kill them. But the tumor cells are sneaky. They have mechanisms of inhibiting the T-cells, and that's where the PD-L1, PD-1 pathway comes into play. So PD-L1 is the ligand. There's also PD-L2. And PD-L1 is expressed on the surface of tumor cells, and that's where we can visualize that and see it and quantitate that with immunohistochemistry, and that's the FDA-approved biomarkers for PD-L1. But PD-L1 expression on the tumor cells will interact with the PD-1 receptor on activated T-cells and suppress T-cells, and it helps the tumor evade the immune system. And so there are, again, FDA-approved drugs, such as pembrolizumab and nivolumab, that inhibit this pathway. They are PD-1 inhibitors. There are also PD-L1 inhibitors that are in clinical trials. Next slide. So the challenge in the field, because not all patients actually will respond to immunotherapy, and there are some toxic side effects from immunotherapy, the challenge in the field is coming up with biomarkers that predict who will benefit the most from these agents. They're expensive agents and, again, have toxic side effects, and we would like to minimize the use on patients who aren't going to respond. Again, I'm not an immunologist. The immune system is very complicated. There are a number of review articles written by immunologists and oncologists that address this issue. I would refer our audience to an issue of Science magazine from May that has a number of articles addressing this. And there's a quick editorial by some leaders in the field, and the title of that editorial is called The Cancer Immunogram. I would refer them to that. But just briefly let me touch on some of the really interesting data out there. So we talked about mutations leading to tumor antigens, neoantigens. Well, it turns out, and there's some excellent data in melanoma, that overall global mutation load or neoantigen load can correlate with who will respond to either anti-CTLA-4 therapy or anti-PD-1 therapy. So that's really excellent news, and it's definitely global mutation load is going to be something we need to keep an eye on in terms of biomarker development. What was also very interesting is scientists had really hoped that by sequencing neoantigens in many patients who are receiving these therapy, that recurrent neoantigens that were common among patients that correlated with response would be found, but unfortunately that was not the case. Nevertheless, overall mutation load has persisted both in melanoma and lung as correlating with response. Next slide. And I would refer you to, there are several papers actually in lung cancer that show this, that the non-synonymous mutation burden that is associated with objective response and clinical benefit and progression-free survival in patients treated with anti-PD-1 therapy. And again, this concept of mutation load, mutations being the neoantigens, there are multiple ways to interrogate. I would say mutation load is a surrogate for neoantigen load, and you can measure neoantigens by RNA transcriptome data. You can use a surrogate, which would be mutation loads by DNA. The tumors types that have the highest global mutation load appear to be lung and melanoma cancers, as well as a couple of other types. But that may be why we're seeing great responses in these patient populations. Next slide. So the question has come up, can NGS or gene panels substitute for whole exome sequencing or transcriptome sequencing in terms of biomarkers in predicting who's going to respond to immunotherapy? Well, there is some data out there that NGS panels and the mutation load detected in these panels do correlate with clinical responses and have some predictive power that is comparable to whole exome sequencing. But I would say there's a lot more studies that are needed. In general, we don't really have any standards for how this, for the wet chemistry, for how these assays are done. We don't have any standards for the bioinformatic pipelines. We don't know what the absolute thresholds are for high, intermediate, or low mutation burden, and how that exactly correlates with predictive power. So there's a lot of work that still needs to be done. Next slide. And this is just another set of data that basically suggests that comprehensive genomic profiling can predict response to immunotherapy. I would say that what I have observed is that, and the literature reflects this, that global mutation load and PD-L1 expression are not the only biomarkers that appear to correlate with responses. We know from a variety of publications that certain oncogenic mutations will activate PD-L1 expression, for example, and influence the patient's response to immunotherapy. We know that resistance mutations can develop an interferon gamma signaling pathway that will inform how a patient is going to respond. And so certainly NGS, whether it's whole exome sequencing or gene panels, can be informative. But How all that comes together with other data on, for example, tumor-infiltrating lymphocytes, what populations of immune cells are present, and other cytolytic expression markers that have been linked to response to immunotherapy, how all that is going to come together in a cohesive story and that we can package into a biomarker panel is still yet to be determined. Next slide.
Great. Thanks, Ali. So I'm going to now open up the Q&A part of this segment, and we'll get right to the first question. And these questions came in as part of a pre-webinar survey. So can you comment on the limitations of circulating tumor DNA testing with regard to clinical setting? And you covered a little bit of this in the introductory slides, but perhaps for specific tumor types. And also assay validation characteristics like lower limit of detection, as expressed by either allele frequency or number of copies and sensitivity.
Sure. I mean, these are difficult. These are important questions, but they are difficult to answer. The challenges of the cell-free DNA is that, first of all, the amount of tumor cell-free DNA that is in every patient is going to be highly variable. And that variation depends on tumor burden in the patient and how well the tumor is diffused. It might depend on tumor type. It looks like in glioblastoma, it may be harder to detect because of the blood-brain barrier, perhaps. It may be harder to detect cell-free DNA sometimes. It also can depend on the contribution of the healthy cell population to the amount of cell-free DNA that's mixed with tumor DNA in the peripheral blood. And so you could imagine that if you have a patient who is in the midst of a massive inflammatory response, that there's going to be a lot more shedding of cell-free DNA from non-tumor cells, and that's going to dilute the mutant fractions from somatic events. So really, in terms of assay validations, anyone who attempts this is going to have to validate their assay over a wide range of input of DNA. And the lower limit of detection will vary over the range of inputs and what's going on in that patient. And there are some simple ways to think about this. You can kind of estimate what your lower limits of detection might be. And we know, for example, that genomic loci are represented in 1 nanogram of DNA. There's about 330 copies of each genomic loci. So 10 nanograms, in our experience, 10 nanograms of DNA is pretty reproducible extraction yields from cell-free DNA from cancer patients. And that would be about 3,300 copies of DNA. If you had 100% conversion of all of those copies in your assay with either the PCR prep or the library prep or whatever technique you're using, you would get 3,300 copies. Well, if you want to detect a 0.1% mutant allele fraction, that would be calling unique reads in 3 copies. So, this is an example of how the amount of input you're starting with can really vastly affect your sensitivity and your limit of detection. But again, anyone who wants to validate these tests are really going to have to validate a wide variety of, a wide range of DNA inputs. So the other thing that's a real challenge is the size of the DNA fragments. Recovery of those very small DNA fragments, like 140 plus base pairs, is technically challenging and probably where a lot of the effort needs to be invested in a development and validation. And I think, Rakesh, we may have a question coming up that's related to this in the next slide, perhaps.
Yeah, that's right. So, I think we could talk about the experimental methods, and I think you hit on size selection, and I think you could expand on that here, as well as the molecular barcode issue that you brought up.
Yeah, so what experimental methods are recommended for purification? Well, I understand from my colleagues who are true nucleic acid biochemists, they tell me that, frankly, the QIAGEN kit for circulating nucleic acid is probably the best kit for extractions. They've tried the automated methods, and they haven't been so good. So I think their recommendation is right now this QIAGEN kit. I don't generally want to try to promote any one company, but I would have to say that would be their favorite. In terms of library prep, I think the most important concept here, and again, I'm hearing this from my colleagues who are nucleic acid biochemists who design these assays, and the really smart bioinformaticians that they work with, is that the wet chemistry of the library prep and the bioinformatics pipeline need to be developed together. And this is where those barcodes come in. So there's this concept, this term barcode molecular families. These barcodes have to be added as part of the library prep. And those barcodes are really important for eliminating errors and false positives. And that's probably the most important point in addressing this in terms of maximizing sensitivity and specificity.
Great, that's perfect. And I think the reference that you put in the initial slide, use the molecular barcode family to really increase sensitivity and also reduced or polished error rates that were normally seen as part of oxidative damage in the library prep. So I think both methods, at least in that manuscript, worked well. So the last question is really, what are the regulatory requirements for cell-free DNA testing? We can limit this to circulating tumor DNA testing, but I suspect the answer will be the same.
Yeah, and I actually had to call my colleague, Dr. Volkerding, on this. I am not aware of any regulatory requirements. I think that's to be determined, at least for circulating tumor DNA. I know that the FDA, for example, and CAP have an interest in regulatory requirements for NIPT, so prenatal testing, and Dr. Volkerding can comment on that a little bit more. Would you like to speak up, Carl?
Yes, can you hear me? Yes, hi, this is Carl Volkerding. Currently, through the College of American Pathologists, there are laboratory requirements that are articulated in a series of documents for that laboratories need to meet in terms of checklist requirements if they're performing non-invasive prenatal testing on circulating cell-free DNA, but specifically for the detection of fetal aneuploidy. At this point in time, there are not yet requirements through the College of American Pathologists for laboratories performing cell-free DNA specifically, but they are in the process, the respective sort of subspecialists within the College of American Pathology committees are beginning to look at this topic because it is an emerging topic, specifically what types of controls and requirements would laboratories need to meet if they're employing cell-free DNA for clinical diagnostic purposes.
Perfect. Thanks, Carl. So I want to spend just a few minutes on the immunotherapy questions. And this slide really summarizes the majority of the scientific or clinical questions. So can you comment on the assay performance when you're using NGS to make immunotherapy decisions? So for example, is there published reliability and error rate? And how does this compare to immunohistochemistry?
Yeah, I actually don't think there's adequate data, published data yet on using NGS to make actual clinical decisions. I think that this is still in the research field. In tumor boards, we talk about this. You know, we may have a report in front of us that, you know, the lab performing the test has commented on this. You know, this appears to be a hypermutated tumor. However, I would just remind everybody that we really don't have enough data yet to know what those thresholds, those cutoffs really should be. And so we're kind of feeling around in the dark at this point. In contrast, for immunohistochemistry, we have 2 FDA-approved assays. And one of them was performed in clinical trials, the 22C3 biomarker for pembrolizumab. That performed well enough, at least in lung cancer, to actually make it as a companion diagnostic test. Is it perfect? No. It's actually very difficult to interpret. You know, the FDA and the pharmaceutical company and DACO have put forth a test through clinical trials that is a subjective assessment. You know, you're using immunohistochemistry to to estimate quantitative levels of PD-L1 expression on tumor cells, and that's a challenge. And I would say I don't think that that's going to end up in the long run being the only test we have available, and I wouldn't be surprised to see it combined with NGS-based assay and other assays that look at expression levels of cytolytic pathways and antigen presentation pathways. I think all of that is coming, and it'll be really interesting to see what combinations of different interrogations and different modalities are used to come up with tests that are more informative than what we have now. But in terms of NGS, the data is very much emerging, and we just need to keep tabs on this.
Great. Thanks so much, Allie. I think we're going to stop that first segment. You know, I want to move on to the second component. You know, our experience at Parian has been very similar with our partners. They're starting to ask about more and more comprehensive assays, including those that use circulating tumor DNA testing or looking at tumor mutation load. And I think this will be a ripe area in 2017. And I really expect our partner sharing network, where we're able to amalgamate data across customers to perhaps feed into the knowledge that's gained by looking at targeted panels and predicting tumor mutation load. So with that, I really want to move to the second segment. I want to introduce Carl Volkerding from the University of Utah and Area UP Laboratories. Carl will be discussing proficiency testing for clinical NGS applications. Carl?
Yes, hi, good morning and good afternoon. I want to first just provide for the audience some general background. Clinical laboratories specifically in the United States that provide clinical testing are required to be certified through ultimately the federal branch of our government through a legislation that was passed in 1988 referred to as the Clinical Laboratory Amendments of 1988, often just referred to as CLIA 1988 or CLIA 88. And this is a requirement. There are several sub-requirements within the CLIA legislation, and one of which includes that CLIA-certified laboratories are required to participate in proficiency testing for the tests that they offer for clinical diagnostic purposes. And essentially, proficiency testing is the external performance indicator of the laboratory's quality and ability to accurately generate diagnostic results. Now, through the years, one of the organizations that has come to the forefront in terms of providing proficiency testing has been the College of American Pathologists. And the College of American Pathologists, or CAP, has what is referred to as deemed status through the Center for Medicare and Medicaid Services. And the Center for Medicare and Medicaid Services oversee administratively the CLIA program. And so the CAP, through this deemed status, has the ability to accredit laboratories. That is, if a laboratory is set up and it wants to offer diagnostic testing, it needs to undergo an accreditation process and then subsequently participate in proficiency testing. And so the CAP both accredits laboratories and administers proficiency testing for a wide variety of diagnostic testing, inclusive of clinical chemistry assays to immunoassays to mass spectrometry-based assays to a variety of genetic-based assays. Next slide, please. So one of the critical challenges that's perennially present in proficiency testing is how to actually develop the material, the actual samples that will be sent to clinical laboratories for proficiency testing. And this is very true for all areas of testing, and it's been especially challenging to develop these types of materials and samples to provide proficiency testing for clinical next-generation sequencing-based diagnostic testing. And this is further challenged by a very rapid and growing diversity of the different types of diagnostic assays that are being based on next-generation sequencing. The first assays that were being offered by clinical laboratories included those that were focused on the detection of germline variants for inherited disorders. This area has expanded from single gene-based testing by next-generation sequencing through multi-gene panels now to exome and whole genome. So this has expanded considerably over the past 5 years. Also within that time frame has been the introduction by multiple laboratories of gene panels for the detection of somatic variants in both solid tumor and hematologic malignancies. And relevant to the presentation by Dr. Grossman, I think we are going to see on the near-term horizon more and more exome and or genome-based sequencing on solid tumors and hematologic malignancies, both for diagnostic purposes of the primary genetic variation, but also to provide these types of information for correlation of neoantigen load as reflected by sequence variation at the genomic level. Next slide. So in order to develop proficiency testing materials, or essentially samples that would be sent to laboratories in a blinded fashion that they would then process through their laboratory protocol and report back results to the College of American Pathologists. Specifically for next-generation sequencing, we have been leveraging the concept of a methods-based proficiency testing. And this is really to assess the laboratory's ability to detect a spectrum of germline or somatic variants in clinically relevant genes. And this reflects the primary analytical goal of this type of testing. Historically, most proficiency testing in the area of genetic applications was what we referred to as analyte specific. So for example, a sample would be specifically tested for the cystic fibrosis gene, with the cystic fibrosis gene being the analyte. Or we would send out samples specifically for KRAS or NRAS testing, with those two genes being respectively analytes. The challenge is when a lab has a 50 to several hundred gene panel for somatic variation, you are now dealing with a diversity of genes, a diversity of variant spectrum from single nucleotides to insertions and deletions. So you're moved beyond, the main question is, can the laboratory detect the variation as opposed to any one specific variation in a specific analyte? So you have to have a broad test to determine broader capability of the assay, hence the concept of the methods-based proficiency testing. This can be augmented by inclusion of samples that have specific variants that you want to make certain the laboratory can absolutely detect. For example, a mutational hotspot in a somatic variant mutational hotspot. Next slide. So we undergo an extensive process to develop proficiency testing materials for next-generation sequencing. For example, in the area of germline variants, we have focused on taking human cell lines that have been appropriately consented by the donor and then characterizing them by subjecting them to exome or whole genome sequencing or both, and then doing a very thorough bioinformatic characterization to essentially identify the variants that are present in the sample. Then that provides us a population of sequence variation from which we can establish a proficiency test. In the area of somatic variant proficiency testing, one of the challenges we have there is to try to essentially replicate the variable or varying allele fraction or percentage of somatic variants at different levels. For example, a 50% allele fraction of a variant versus a 5% allele fraction, because this is the type of range that laboratories are actually diagnostically testing for. So therefore, you need to both test the laboratory for its ability to detect a variant, but also to detect the variant at a relatively low allele fraction. So the lower limit of detection of the laboratory needs to be assessed as well. And in this case, we're utilizing cell lines that have can be genetically engineered specifically to reflect those types of variations in both sequence and allele fractions. Next slide. So in 2005, the College of American Pathologists launched the first proficiency testing for next-generation sequencing, and this was specifically a program or a proficiency testing program that would encompass laboratories performing both gene panel testing for a variety of inherited disorders to exome and genome sequencing. And so this particular proficiency testing program is characterized by laboratories receiving 10 micrograms of extracted DNA from this highly characterized cell line, and then the laboratory receives a list of information of up to 200 chromosomal positions that they can interrogate based on, depending upon their assay. So perhaps a laboratory is doing a multi-gene panel for a particular disorder, and in the 200 selected chromosomal positions, they may actually test for 7 or 8 of those positions. So that's what they focus on. They perform their assay using the DNA, take it all the way through their bioinformatics pipeline, identify variants, and then report back to CAP any variation, or if the chromosomal position is a reference or wild-type position, they report that information back to CAP. Is a variant detected or not? What is the zygosity of the variant? And then if a variant is present, they're requested to describe the variant using standardized nomenclature from the Human Genome Variation Society. And this particular program has been in effect since 2005, and now we're in almost at the end of 2016. Next slide. Then subsequently, 2 additional programs for proficiency were introduced specifically for somatic variants. And in the top panel of this slide, it just shows the catalog description of one of the 2 programs. And this first one at top is specifically for laboratories performing solid tumors. And you can see in the additional information below in the paragraph, you can see the list of genes that this particular proficiency testing program is relevant to and will assess. And this particular program, laboratories receive physical DNA samples. They are asked to process them through their sequencing platform, targeted enrichment, for example, followed by their bioinformatics, and then again to report back to CAP what they've identified, the zygosity, and what is the description of the variant. In the lower panel is essentially a correlate to this, which is a proficiency testing program for a variety of genes that are involved in hematologic malignancies. And you'll see those genes listed in the additional information. And it's important to note that biologically there is overlap in some of the genes between the solid tumor panel proficiency testing and the hematologic malignancies. For example, P53, KIT, for example. And one thing additional on this is that proficiency testing is usually administered twice per year, so you'll see on the right-hand side it says program information, the amount of DNA, how many samples, and then in this case shipped out twice per year for laboratories so that they can meet their CLIA requirements. Next slide. So we do have, though, unfortunately, we have some very critical limitations of physical proficiency testing materials. One, they're very expensive to produce and maintain, especially cell line-based material. They have a genomic limitation of information, if you will, so you ultimately cannot replicate the diverse spectrum of variants that a laboratory may encounter during clinical diagnostic practice. And so any one sample will be limited in terms of the type of variation that is present in the sample. And also, these are limitations, so one approach that we're taking to this limitation is actually to turn to what we're referring to as in silico proficiency testing, which is based on taking DNA sequence files and actually introducing pathogenic variants or variants of uncertain significance, perhaps, depending on the scenario, into the sequence files, and then returning those, providing those to the laboratory, and then they can assess their bioinformatics pipeline. Now, there are a lot of variations on the theme that could potentially be pursued by this, but in the next slide, I will discuss the first in silico proficiency testing product that we have available for somatic variants. The approach we took on this is to focus on 2 popular commercially available cancer panels. One offered by Illumina, the TruSeq Amplicon Cancer Panel, one offered by Thermo Fisher Ion Torrent, which is the AmpliSeq Cancer Hotspot Panel. And in this setting, these are what is provided to the laboratory and it's important to note that the laboratory has to be performing either of these two assays for this proficiency testing to be relevant to them, is that they receive sequence files, either FASTQ files and or BAM files, and within those files a variety of variation has been introduced into the relevant genes that are captured by these methodologies, captured and interrogated, and then the laboratory takes these FASTQ and or BAM files and then introduces them into their bioinformatics pipeline, takes that through their bioinformatics process, identifies the variation, and reports that back to CAP. And so these are programs that are now ongoing, and there will be more to come in the next year and onward from CAP, and again, to try to meet the diversity of the growing applications. Next slide.
Right, so Carl, I want to thank you for the good primer and introduction into proficiency testing, as well as that for NGS, and it's great to hear about these first 4 products. I'm going to jump right into the first question. So far, the proficiency products that have been released by CAP have been educational in nature. Can you talk about when this will become graded, and can you also talk about in silico products that go beyond the defined assays that you described in the previous slide?
Sure. Thank you for that question. So when the process to develop a proficiency testing typically follows, and then the launch typically follows this format. There's the idea and conception of what would be the proficiency testing product, as generated by individuals that do volunteer work and serve on committees for the college. And then essentially a pilot project is conducted based on the concept to see if it can be successfully piloted through volunteer laboratories, a small subset of volunteer laboratories. And then, if that looks promising, it is then launched as an educational proficiency testing that laboratories can subscribe to. Important to appreciate in educational proficiency testing is that the laboratories that participate are not graded specifically, and therefore they will not be penalized based on their performance for that proficiency testing. The CAP reviews these educational proficiency testing surveys. They typically occur, they always occur in the first year of the launch of a proficiency testing new program. It can certainly extend into a second year until there's sufficient data accumulated by CAP in terms of how are the laboratories handling this new PT product. Are they technically being successful in handling it logistically? Are the results that are coming back in sufficiently robust that it warrants that we can convert this to a graded proficiency testing program? Of the 4 products that I just described, the germline product that was first launched for germline detection of panels within panels, exomes, or genomes has been converted over to a formally graded program at this point in time. The other 3 programs are still in their educational phase. Now, in terms of what's next, specifically with regard to in silico proficiency testing, what we are trying to work for, our goal is to work for further developing what I would refer to as our NGS bioinformatics infrastructure within CAP to allow us to do truly customized in silico proficiency testing. What I mean by truly customized is that a laboratory in the future would submit their sequence data files generated through their own physical samples to CAP. those data files would then be in silico mutagenized and then sent back to the recipient laboratory. So they would either submit data files from samples that they had sequenced, or CAP would provide them a sample, a physical DNA sample to sequence, and then they would submit those. So we're looking at different options in that regard. And then the real value of this kind of customized is that a laboratory might have a 200-gene panel for a specific area of inherited disorders that they want to be assessed on, and we would know what those, they would provide us the genes, they would provide us information about their technical sequencing. We would receive that information, and then we would introduce sequence variant content into those files that would be returned back to the laboratory. That way we can begin to accommodate the wide diversity of assays that are being utilized by different laboratories, everything from different size somatic variant cancer panels to different germline panels to exome and genome. So that's what's on the horizon over the next several years.
Fantastic, Carl, and I think this matches really well with our own experience at Parian, where even within a small subset of partners, we're seeing over 30 unique assays, many of them customized, and so I think the custom in silico PT is really going to be of value to organizations like us and labs that participate in clinical NGS. You know, with that, I want to now hand the conversation over to Julia to manage the questions that were posed by audience members during the webinar. Julia?
Thank you, Rakesh. So just before we go into the Q&A, we'd just like to ask our attendees to take a moment after the webinar has ended to take our exit survey to provide us with your feedback. So now let's start with the general Q&A. So here's a question for Ali. How does tumor heterogeneity complicate the use of liquid biopsies for diagnostics?
I think that's an excellent question, another very difficult question to answer. Tumor heterogeneity will always be a thorn in our side, no matter what kind of assay we're doing, whether we're testing directly or indirectly. The hope is that what is circulating in the blood would be more representative of, you know, the whole burden of the tumor that a patient may be harboring. You know, there certainly is data out there of high but limited concordance of mutation between primary tumor and metastasis and different metastasis. You know, I think we're going to still have a problem of tumor heterogeneity with liquid biopsies. Again, you could have some sites of disease, brain metastasis, for example, that you know, if a patient has a lung, liver, and brain metastasis, what we see in the blood may only represent what's in the liver and the lung because of the blood-brain barrier issues and clonal heterogeneity of metastasis. I think the data is you know, still emerging on that, and we need a lot more studies. But I anticipate tumor heterogeneity is always going to be a challenge.
All right, thank you. Another question for Ali. What is the difference in terms of clinical utility between liquid biopsy and solid tumor testing for identifying germline mutations?
For identifying, I'm assuming that the question pertains to germline variants that may be pathogenic, that might be identified in tumor normal paired sequencing, I'm guessing.
Yeah, I guess so too.
Yeah, and there's probably many ways to interpret this question. So again, I honestly don't think there's enough data on that yet. You know, in cell-free DNA, you can certainly see different fractions. So cell-free DNA from normal healthy cells, the data is beginning to show that those are a little bit larger fragments, maybe around 160 base pair, whereas tumor DNA may be closer to 140, but you'd have to have a way of distinguishing germline from, maybe that's going to be size, maybe that's going to be methylation patterns, but you would have to have a way of distinguishing germline from, without directly interrogating the buffy coat. You could do that with a blood sample. You can split out plasma from a buffy coat and pair them that way, and that way you'd have a lot more confidence about what you're sequencing. But the ability to distinguish cell-free DNA from tumor from healthy normal cells that are also populating cell-free DNA is still emerging. And you know, we have this issue with directly testing the tumor as well. The only way to be truly confident about a germline variant that you might be suspicious of when you've tested the tumor alone is to actually go back to either simultaneously or retrospectively go back after the fact and evaluate the patient's germline with a separate sample that's non-tumor. I hope that answers the question.
Yeah, thank you, Ali. Unfortunately, this is actually all the time we have today for questions. So with that, I'd like to thank our roundtable participants, Ali Grossman, Carl Volkerding, and Rakesh Nagarajan, and also our sponsor, Perion DX. And if we didn't have time to get to your question, we will try to have one of the panelists answered after the webinar. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. So thank you very much for joining us today for this genome webinar.